From GCSE, you should be familiar with the concept of a monohybrid cross, even if you've never used that term before. Basically, when we inherit genetic information, when we inherit genes from our parents, we inherit one allele for that gene from each parent. So you should remember that there were a number of different combinations of these alleles. You can have two dominant alleles, one dominant and one recessive allele, or two recessive alleles. We refer to the genotypes that have two of the same allele as being homozygous. This one is homozygous dominant, this one is homozygous recessive. The one that has one dominant allele and one recessive allele is a heterozygote. Dominant alleles only require there to be one copy of that allele in order to be expressed. So heterozygotes commonly have the same phenotype, the same physical or chemical expression of that gene as the homozygous dominant. When we look at trying to work out what the phenotype is of an organism, or when we're trying to study what the genotype is, we commonly use things called test crosses. And I think it's quite important that we look at the idea of a test cross first. With a test cross, you have to take two true breeding individuals. True breeding individuals are homozygous for as many genes as possible at all of their loci. So you should find that a true breeding organism is either homozygous dominant or homozygous recessive. And this is because generally they tend to have been inbred, so that you have effectively a doubling up of the alleles. It's very easy to do with plants. Many plants will self-fertilize easily. It's a little more interesting trying to do it with animals. So we would normally take two true breeding individuals, and they would normally be homozygous dominant and homozygous recessive for a particular trait. Now, these will all form here on this side of the cross gametes containing the dominant A allele. All of these individuals' gametes will be recessive A alleles. And so you should end up with an entire generation that are all heterozygous. We refer to this true breeding generation as being the P for parental, and the first generation from that cross, the first filial generation, is F1. This is where the more interesting parts of doing test crosses comes in, because the next stage is to allow the F1 generation to self-fertilise, or for two individuals of the F1 generation to be crossed with each other. Here is our F1 generation. We've got one individual with dominant and recessive allele. We have another individual with dominant and recessive allele. Now you can here look at all of the possibilities for the individuals by drawing a genetic diagram. So you can have your four possible outcomes of the cross here, and you can show this one, dominant allele, going into these two. And you can show this allele going into these two. And then we look at all of the possibilities here, because we've got a big A, got a big A, there's a little A, there's a little A. Okay, we can have this dominant A allele coming into this zygote. We can have it going into this one, and we can have this arrangement of the recessive alleles. So we end up with a homozygous dominant, a heterozygote, another heterozygote, and a homozygote recessive. This becomes really confusing, And by far the best way of showing these crosses is with the Punnett square. So rather than splitting them up like this and showing the gametes going into different zygotes, we separate out the gametes from each parent into the column headings or the row headings here. So we have a big A, dominant allele, we have a little a, big A, little a, and then you look at the product in here, we've got a big A and a big A, so we've got a homozygous dominant. Here we have a heterozygote, here is another heterozygote, and here is a homozygous recessive individual. And if you were then looking at the phenotype, then you would find that these have the dominant phenotype, and this one has the recessive phenotype. So the ratio of dominant to recessive phenotypes in a monohybrid test cross of this nature would be 3 dominant to 1 recessive. And this is referred to as a Mendelian ratio because it follows this strict principle of Mendelian inheritance, that alleles are segregated separately into the gametes, and there are a full range of possible recombinations of these alleles. You sometimes get test crosses that are a little different. Let's look at a test cross between a member of the first filial generation and the true breeding recessive form of the organism. If we look at our Punnett square, I'll do it a bit smaller, we'll have a big A, dominant allele here, and a recessive allele here. And then this parent contributes only the recessive alleles. So we get this arrangement. Now you can see here that these two will both express the dominant phenotype. These two will express the recessive phenotype. So here, where we've got a heterozygote crossed with the homozygous recessive individual, we'll have a Mendelian ratio of 1:1 of dominant and recessive alleles. So this is something you should look out for. As a related note, you should also be aware of incomplete dominance or codominance. Codominance is probably a little bit too much for this particular episode, but incomplete dominance would give you a separate phenotype for the heterozygote. And a great example of that could be flower colour. And this is because in the heterozygote you have one allele that produces, say, pigment colour. If you look at the homozygous dominant individual, they have 2 copies of those alleles, so can produce twice as much pigment. This can often lead to homozygous dominant plants being, say, red, heterozygotes being pink, and homozygous recessive plants being white. So in those cases, you would see, in the case of a regular heterozygote test cross between 2 pink individuals, you would see a 1:2:1 ratio, because you've got one phenotype for the homozygous dominant, one for the heterozygote, and one for the homozygous recessive. Now we need to look at something a bit more complicated. Dihybrid cross takes 2 genes at different loci and looks at the outcome of crossing those individuals together. So we're looking at individuals who have gene A and gene B. If we were to take 2 true breeding parents, then what we usually have is one that is homozygous dominant for both genes, crossed with one that is homozygous recessive with respect to both genes. This is how we express it. We put the 2 alleles of one gene together and the 2 of the other together. Now, this means that we've got a whole load here of parental alleles, big A, crossing with the little a. We've got 2 copies of big B, the dominant, crossing with 2 copies of little b, the recessive. This is quite an easy one to work out. All of your F1s will be heterozygous with respect to both genes. This is where the complicated bit comes in, and this is where you will see why I haven't drawn the genetic diagram showing the arrows pointing where the gametes are going into the zygote. This becomes extremely difficult to visualize. So with our F2 generation, we're going to be crossing or self-fertilizing the F1 hybrids. There's a way we can do that, and you saw the Punnett square earlier. We're going to use exactly the same sort of Punnett square, except this one, rather than being a 2x2, is a 4x4 to take account of the fact we've got 2 genes that we're looking at. If you were doing a trihybrid cross, then you would have to have an 8x8 Punnett square. Fortunately, that is not on your syllabus. Here is a Punnett square that I've drawn, and I'm going to show all of the possible combinations of gametes that you may have given these F1 test cross. So here we will have one big A, or dominant B. We could also have a dominant A, recessive B. We could have a recessive A, dominant B, and recessive A, recessive B. Same down this side. Now, just as before, I'll take a different pen for this, we're going to go through and look at what the product is of the combination of the alleles in this row and this column. And we'll see we're getting a double big A, double dominant here, and dominant with respect to B as well. I'm going to fill the rest of these in really quickly. If you're struggling to fill in these Punnett squares, then a useful trick is to remember that all the way along this diagonal you've got a symmetry. So you've got the same combinations of alleles here, 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 and here. Okay. Now we need to look at the phenotypes of each of these. So we've got, let's assign some properties to this. Let's assume that we're looking at gene A, which controls the height of the plant. So dominant A is tall and the recessive A is the small allele. And so we've got, let me just write a note of that. So A is tall, little a is a dwarf, big B, let's say this is a nice blue flower, and little b are white flowers. So if we look at the genotype here, we've got both the dominant allele, so it's going to be tall, and it's also going to be blue. Here, again, another tall blue. I'll fill in all of the other tall blue plants now. And now I've done that, you should be able to see we've got 1, 2, 3, 4, 5, 6, 7, 8, 9 tall blue plants. So remember that, 9. Now we need to look at what the phenotypes of the rest of these plants are. So here we've got tall, but it's got 2 copies of the recessive alleles, so it will be white. Again here, and down here. These 3 will all be dwarf blue plants. And this one alone will be a dwarf white plant. Let's see if I can actually write this in here. Well, assuming you can read that, we have got 9 tall blue plants 3 tall white plants, 3 dwarf blue plants, and 1 dwarf white plant. With a dihybrid cross, we have 4 phenotypes available, whereas in the monohybrid we had 2. And where the Mendelian ratio for the monohybrid cross was 3:1 when you cross 2 heterozygotes, here the ratio will be 9:3 to 3:1. Reviewing these phenotypes, we had tall blue, tall white, dwarf blue, and dwarf white in the ratio 9:3 to 3:1. Again, these are the Mendelian ratios for a cross like this. If you were to cross a heterozygote from the F1 generation with the homozygous recessive true breeding parental generation, then your ratios for this, in the same way that you had 1:1 heterozygote to homozygous recessive, you would end up with a 1:1 to 1:1 ratio. It could get extremely difficult if you're looking at incomplete dominance in a dihybrid cross, but you should not be asked to do that on A-level exams. Sometimes these ratios don't quite pan out. You can do 1,000 test crosses and you get results that do not appear to fit the Mendelian ratios. And there could be a number of reasons for it. The first reason for this could be something called gene linkage. If you look at a chromosome, so here's a chromosome, here's a homologous pair of chromosomes. If you were to look at a gene locus here, let's say this is A, and then let's look at a gene locus B up here. Okay. So of course you'd have big B here, dominant B, and dominant A here, and likewise recessive on the other side, because these are identical, they are sister chromatids. When you've looked at meiosis, the process of forming the gametes, you have looked at the idea of crossing over. If there is crossing over that occurs here on this arm of the chromosome, then you'll find that this chromosome that otherwise has the dominant B allele, rather than having dominant A, will have the recessive A allele, and vice versa. If, however, instead of B being up here on a different arm of the chromosome, if it was right in here, let's do B dash, then when crossing over occurred, it would be very unlikely that it would occur right between the locus for A and B, in which case those gene loci would become linked together. So even when crossing over occurs, they are not necessarily inherited independently of each other. So in the event of locus B being so close to locus A, you would find that whatever the allele combinations were on those chromosomes, that would be how they would be inherited. So if here you've got dominant A allele and dominant B allele very close together, then you would not see the allele combinations, say little a, big B, and big A, little b. That would shift everything in favour of the big A, big B, and little a, little b alleles being in the gene pool here. So you would expect to see very few of the 3 and the 3 part of the Mendelian ratio, and more of the 9 and the 1 part of the Mendelian ratio. Okay. To ascertain whether there is gene linkage, you can do a chi-squared test, assuming the Mendelian ratios are your expected values, and what you've got out of a possible linkage scenario is your observed values. But that would be a different video completely. The final thing that I would like to talk to you about in regards to the dihybrid crosses is the idea that many genes and many alleles at different loci may affect the overall phenotype. And so we're going to look at this from the perspective, rather than say the allele that being transcribed and translated produces a pigment that it is itself present, say a pigment colour. Let's look at the idea of the alleles transcribing and translating to form enzymes that catalyse different stages of a metabolic process. So looking at pigment is a good way to go with this. Let's say we have a precursor going to intermediate to a product. This reaction is catalysed by enzyme A, and this reaction is catalysed by enzyme B. Okay, let's assume that it's a product. So precursors here for pigment tend to be colourless, as often are intermediates. And then let's say this product is a red pigment. So if we were looking at our dihybrid crosses, there are a number of distinct genotypes that we can look at. So the first being the one that is homozygous dominant with respect to both genes, we can refer to as being big A, and I usually put an underscore here just to indicate it doesn't matter whether this is dominant A or recessive A, and dominant B. Same scenario here, it does not matter whether this is a dominant B or a recessive B, the dominant phenotype will be expressed. Then we have the option that we've got the recessive condition for A, but the dominant for B. That we've got the dominant condition for A and the recessive for B, and then finally being recessive with respect to both genes. So here we can look at what happens in the event that the recessive alleles are present. Now, if dominant A is an allele that codes for an active functional copy of the enzyme A that catalyses the precursor to the intermediate reaction, then if there is a recessive homozygous genotype, enzyme A will not be produced. Same with enzyme B. If you've got the recessive genotype here, a functional copy of enzyme B will not be produced. So if we have dominant A and dominant B, we should have 9 of these. Here are our Mendelian ratios again. Then these are going to be, we'll have the intermediate produced and you'll have the product. So they will be red. Okay. If you have the recessive trait at A, then no intermediate is produced, so they're colourless. So if a flower colours, then they'll be white. We have the dominant allele here at A, so the intermediate is produced, but the product is not because the recessive condition is at B. So these are also going to be white. And then here, nothing's going to happen at all. The flowers will be white. So this part of the Mendelian ratio, the 3 to 3 to 1, we combine it all together and we end up with this modified ratio of 9 to 7. This is an example of complementary epistasis. Other Mendelian ratios that show different types of epistasis. And so a few of these that you should remember, as well as 9 to 7, you should be able to recall 13 to 3. We occasionally have 9 to 3 to 4. And you will occasionally see 12 to 3 to 1. Okay? So any of those modifications of a Mendelian ratio there, if they fit within the 9 to 3 to 3 to 1, then you've got epistasis occurring. If you do a chi-square test on it and it doesn't easily fit into these ratios and the chi-square result is significant, then you've got gene linkage. I hope that has been a useful run through. It's really only a very brief discussion of the inheritance aspect of it. I haven't talked about sex-linked inheritance. I haven't talked about mutations. But I hope there was enough from GCSE and other sources to substitute for that. Until next time.